So one of the fundamental problems in biology is how organisms pass genetic information from one generation to the next. Let's take a look at a man with a beard and a woman with long hair who are going to have a kid. Organisms use all sorts of different strategies for passing genetic information, but before we really go into how humans or other organisms do this, let's talk about what genetic information is. So, in general, genetic information is the information that the next generation needs to be able to build a body and survive in the environment. Now, humans keep that genetic information on chromosomes. Chromosomes are a mix of DNA, so one chromosome would have one long molecule strand of double helix DNA. It'll have some proteins, and it'll also have some RNA. So these proteins and RNA help the DNA fold up and fit inside the cell. The DNA would be much too long to fit inside the cell without the proteins and the DNA being there. So the man and the woman each have more than one chromosome. They each actually have 46 chromosomes, and those are 23 pairs. So 23 times 2 equals 46. And that might look a little bit like this, say. So you'll notice that we have 23 pairs. Don't pay too much attention to the colors. I'm just trying to illustrate that while these things are paired up, they are very similar in the type of information that they carry. They actually do carry slightly different information on each member of the pairs. Across the pairs, say the first pair versus the second pair, the information is actually very different. Okay, so each of these parents has their 46 chromosomes. How do they pass those chromosomes on to their kid? So you might think that both of the parents are going to take all of their chromosomes, all of their genetic material, and just hand it over to the kid and let the kid sort it out. And that might look a little something like this, where these are the mother's chromosomes from right here, and these are the father's, and they've just taken them and put them together. And you might look at that and you might sort of see a way that this could possibly make sense, but it turns out that this is not what's going on. So it's not this. What is going on is a special type of cell division called meiosis, which produces germ cells. Germ cells are better known by the names egg and sperm. So what's so special about the egg and the sperm? Well, there are two things. One special thing about eggs and sperm is that they each have one half of the genetic material that's in most of the other cells in the parent's body. The other really special thing about eggs and sperm is that they can give rise to a lot of genetic variation. So how does this whole cutting genetic material in half and genetic variation thing work? Well, what ends up happening is that each parent, so the mother and the father, each give one of each of the pairs of the chromosomes to the kid. So that takes care of the cutting the genetic material in half part. The genetic variation comes in the way that each member of each pair is chosen to go into a specific egg or a specific sperm. And it turns out that it's totally random. So each egg and each sperm gets one member of each pair, but there's no coordination between pairs. So if we're looking over here at the mother's chromosomes, the fact that, for instance, this orange one goes into an egg has no impact on which one of these two in the second spot will go into the same egg. It could be this green one, or it could be this purple one. It's totally random. You've got a 50-50 chance of getting either member of each pair for the mother or the father. Now, because those are the same probabilities as a coin toss, I actually went and found a coin and tossed it before I started doing this video. And tossing the coin told me to use the orange on the left from the mother and this yellow on the right from the father in the first position. In the second position, a third coin toss told me that I should use the green on the left from the mother and the purple on the left from the father. In the third position, a fifth coin toss told me that I should use this pinkish salmon colored one from the mother and this purple one from the left from the father. So I'm going to cheat a little bit and just throw in something that I already had drawn up beforehand, but I did a similar thing where I was taking one member of each pair from each parent and putting it in the position for the kid. So this purple from the mother ended up there, this green from the father ended up here, and so on for the rest of the chromosomes. At this point you might be asking yourself why it's important that the genetic material be cut in half. Sometimes it isn't. So sometimes you end up with an egg or a sperm that starts out with too much genetic material. And one example of when that happens is in a condition called trisomy 21, which is more commonly known as Down syndrome. So I drew this one chromosome in red here in the 21st position, and now I'm putting it over here. So let's say that the father created a sperm that instead of having just one copy of the 21st chromosome had two copies, and so the kid got both copies of the father and still got the one copy from the mother. Trisomy 21 is a really good example of why it's so important for the amount of genetic material to be so tightly controlled. Remember, this genetic material helps the body grow and function by producing proteins, and if you have too much genetic material, you can end up producing too much protein or protein at an inappropriate time, and you can end up with really serious disease states because of that. So anyway, that's why we go through this entire thing of cutting the genetic material in half. Let me just get rid of that extra chromosome there. Now, we've also mentioned genetic variation several times. Genetic variation is important, but it's actually not entirely clear as to why. There are a lot of species that don't have very much genetic variation from one generation to the next, and they actually do really well. For instance, bacteria, in a lot of situations, the daughter cells will look almost exactly like the parent cell. So anyway, humans do this. They create all this genetic variation. They have the genetic material, and they do that again by creating these germ cells that we mentioned earlier. So let's take a look at some of the aspects of what happens to the genetic information when the father's body produces a sperm cell. So let's just take a look at the man's first two chromosomes, sort of to illustrate what's going on. So we're looking at the man, just put a reminder for that there, and we're going to take a look at what happens to these chromosomes as the man's cell goes through meiosis to give off a sperm cell. And I'm just going to draw the cell that these things are sitting in down here. Just remember that these pairs of chromosomes are sitting inside a cell. Before meiosis even starts, these chromosomes have all already replicated. So we start off here, and we go to still a single cell. It's grown a little bit. And now, instead of having just one copy of each of these chromosomes, we've got two, and they're stuck together in the middle like that. At this point, it's kind of useful to think of these replicated pairs of chromosomes as a pair of socks that you folded together after doing laundry. Sort of like that. For now, they're going to move as a unit, but eventually you're going to pull them apart. So in the first real step of meiosis, this cell here splits in two. At this point, the chromosomes undergo independent assortment and go off into the daughter cells. Independent assortment means that we have options for the way that we can distribute these different chromosomes. We could take both of these and put them in one cell and the other two chromosomes into the other cell, or we could take these two chromosomes and put them into one of these cells and put the other two into the other cell. This is what I was doing up here with a coin. Which one goes with which, whether it's the left with the left and the right with the right, or if we mix left and right, is totally random. So let's go with the way that I've got it marked right now, with the yellow and purple going into one cell and the green and the pink going into the other. So let's stop for just a second and take a look at the genetic information that we've got floating around at this point. Right here, the cell is what's called diploid. It's got two versions of each type of chromosome. You'll see this written as 2N, and the cell with the replicated chromosomes is also diploid because even though it has more genetic material, it only has a couple of extra copies, not any actual new information. Once we get up here where the cell is divided, we can say that there's a real difference in the amount of genetic information in each cell. These are what are called haploid, or written 1N, or sometimes just N. So again, notice it only has one version of each chromosome, even though it has two copies of that version. Okay, so going to the next step of meiosis, these daughter cells each divide and give off two cells. And here is where you tear the socks apart. That is, you separate the sister chromatids. So one thing that we should note at this point is that because of this first choice that we made here, we end up with these two types of cells. If we had made the opposite choice by choosing to put these two chromosomes in one cell together, we would have had two totally different types of cells come out here. So there are a total of 4 possible cells. That's because that's equal to 2 to the second. The number in the exponent here is a 2 because we're using 2 chromosomes. Remember that a human has 23 pairs of chromosomes. So a human would be able to generate almost 8.5 million different germ cells. There haven't been any chemical reactions or anything like that going on here. Just random independent assortment. Now remember that this was for the father. The mother does something slightly different. I'm going to work backward from what we got for the father here. This was just copied and pasted in. So where the father produced cells that will generate 4 sperm, the mother doesn't generate 4 cells. Also at this stage, the mother doesn't generate 2 symmetric cells like this. Instead, she generates 1 large cell and 1 small one called the polar body. The polar body doesn't have enough cellular machinery like endoplasmic reticulum and mitochondria to be able to survive, so it dies. The large cell goes on to divide again into another large cell and another polar body. This polar body dies too. At every stage, the mother and father's information is similar. So diploid here, haploid here, and haploid here. The polar bodies are also both haploid. There is one other major source of genetic variation that comes out of meiosis, and it's homologous recombination. Homologous recombination happens early in meiosis. It actually starts while the chromosomes are replicating. The homologous chromosomes, or the members of these pairs that I've been talking about, actually line up inside the cell and cross over each other. And they're physically connected, like this. What happens in homologous recombination is that the chromosomes will actually exchange part of their DNA strands. So at this point here, where the homologous chromosomes touch, they can actually exchange the DNA strand that's part of the chromosome. That might look something like this after the first cell division. Notice that these two sections of the homologous chromosome still carry the same type of information, but it might be a different version. Okay, so to sum up, meiosis is a special type of cell division. Meiosis is a special type of cell division that produces egg and sperm. It cuts the genetic material in half and introduces genetic variation into the population. It cuts the genetic information and material in half by independent assortment of homologous chromosomes and introduces genetic variation the same way. It also introduces genetic variation by homologous recombination, where chromosomes split parts of their DNA strands.